So this talk is about Mcule.com, which is a website where you can do some drug discovery. You can use drug discovery tools. Drug discovery is basically about finding a needle or a few needles in a haystack. And so the question is how to find them. The haystack is usually quite large, and so if you apply virtual screening, what you can do is to reduce the size of the haystack. So you can start with the large one and end up with the small one. You can order them and test them in your lab. So at Mcule.com we provide you with a large haystack, but we also provide you with the tools that can be used to do this transformation. And at the end you can order the most interesting compounds. So in the first part of my talk I will talk about our compound database. Looks like that. So that's our haystack, currently comprising of 3.5 million compounds. And you can browse this database, and if you just click on one specific molecule, you will find yourself on an index page of this molecule. Down here under the tab of product availability, you can see which chemical suppliers are selling this compound, and on the right hand side you can see some identifiers, some basic properties. And yeah, on the first step, the top you see inches. This is because the registration system of Mcule is based on INCHI, the INCHI string. We are also a member of the INCHI Trust. And but it actually turns out that INCHI as itself is not enough for a rigorous registration system, so we complemented it with a lot of structural checks and preparation steps such as valence check. We look for radicals, isotopes. We do novelty check beyond the INCHI rules even, and we correct for stereochemistry and drawing errors and etc. So if a compound passes all these structural checks, then it gets improved and it makes its way into the Mcule database. So I'm going to show you some compounds that haven't been approved. So for example, these two compounds, they are essentially identical. They are tautomeric forms, different tautomeric forms from the same compound. And if you just use an INCHI for a registration system, you will get 2 INCHIs for the 2 forms. Even if you turn on extra tautomeric rules in the INCHI, you will still get these 2 different identifiers. However, these compounds are the same, so they should get the same ID. Right. Another example, quite typical, I would say, is wrong directionality of a wedge bond. So here, this wedge bond points to this nitrogen, which is definitely not a tetrahedral central, but this one is. Actually, we have seen a lot of entries like this, and this is actually because some chemical suppliers using molecular editors don't support the directionality of wedge bond. So we filter all these out and correct for these errors. Another one, this is a valence error here. Probably a charge is missing, a positive charge from this nitrogen, or this double bond should be a single. And yeah, this is a drawing error. You might recognize that here there is a nitrogen and the carbon overlapping each other. So someone wanted to draw a nitrogen here, change a carbon atom to a nitrogen, but both atoms are actually there. And from the INCHI, you see there is a dot, which means that this entry contains 2 fragments. Okay, so here is a fragment and another one. So all these problematic entries can be filtered out. Now this is a nice example. You don't see any problems per se, but if you take a look at the at the inchi, it turns out there is a number 2 here, which means that there are 2 identical molecules in this entry. So what happens was actually this entry contains 2 molecules perfectly overlapping each other. So if you just take a look, you won't see if this is the case. Right. So there are several such structural checks. What we do is, and it's important to mention that actually we do all these structural checks in the registration system at 2 different levels. So we have a compound level here, which is tautomer and protomer independent, and this compound can be associated to different tautomeric forms. We call this the structure level. And so you might recognize that so their IDs are very similar. So basically the first part is the same, and here you have an additional number for the structures, for the specific tautomers, and we just number them consecutively as we find them. If you click on one of the tautomers, you will see that all these structures, these tautomers, have specific index pages as well. And here, if you click on Show Compound, and you can get back to the compound level, actually. So there are these 2 levels, and you can very flexibly change the view between these 2 structural levels. Okay. The other important thing to mention is that if we have a multi-component entry like this one, here we have 3 components, 1, 2, 3, and we register this as a whole, so it gets an Mcule ID, but we also separate them into components, as you can see here, so it contains one of these components and 2 of these. And we register and prepare all these single components, and we will also add a specific component type to them. So they either get a main component type. So in this sample, this is the main component, and you see this is a counterion. We also distinguish between solvents and crystal waters. And adjuvants, such as antioxidants or these kind of things. Right. Yeah, so this is the haystack again, and we want to get this haystack bigger and bigger. So we are looking for other chemical suppliers to add, to integrate their compounds into our database. And what we provide is that we do careful structural curation, and they can basically, on our website, they can share their compounds, they can browse it, they can search it. So we are looking for the integration of other libraries, of other chemical suppliers. Right, so yeah, regarding the second part, which is about searching and screening, we try to integrate this huge haystack again with several screening tools coming from open source resources and also commercial tools. And we try to integrate all these things together, and we use cloud technology, so you get an infinity of computational capacity, and at the end you get the results. So this is how it looks like. You can set a very flexible workflow. Here we have 6 different filters. You can set filter types. You can set all the parameters, and yeah, you can select which collection database you want to screen. You click on screen, and at the end you will get virtual hits. And here comes the third part of this pipeline, that you can basically order these compounds by just a single click. Yeah, after the single click, you can You can specify the amount you want, the required purity, delivery format, delivery time, if you have a preferred supplier, and address, etc. And yeah, you can send this form to us, and we will try to deliver all these compounds to you. So let's see some examples. So on the website there are 3 tabs: search, screen, collections. Search is for very simple searches, like exact, substructure, or similarity searches. Here you can enter an Mcule ID or an INCHI identifier or a SMI string. If you click on load structure, it will appear in ChemWriter, and so you can set a few parameters only, because this is for simple searches, and you can click on search, and here are the results. So the search is performed very quickly, but if it takes some time, you can so if any of the molecules already pass the filters, you will see them appear already. You can change display levels. You can see these molecules in a grid view, and so all these molecules are actually in a collection which you can order or you can export, and as an SD file, for example. But if you just browse them and say that, okay, these 3 molecules are the most interesting ones, you can click on them, and you can click on Add To, so that you can add them to another collection. Here, this is telling you that you haven't created any collection so far, so you can just click on Create a New Collection, add the name, add the small description, and you can choose between 3 levels of privacy. You can create a private collection, which is only for you. You can choose unlisted, which means that only someone who knows a specific link can access to your collection. And there is public, where the collection you create can be seen by anybody. So after you created the collection, see it appears here, and now you can add the compounds. And if you go to collections and select this collection you just created, you will see that these 3 compounds are already here. So now again you can ask for a quote if you want to order them, or you can export them in different formats. So let's move on to screen, which is where you can build a virtual screening pipeline, basically. Here we have two filters, one for similarity searching, one for substructure search. At the moment, as the website is just launched, we have these basic searches. Well, we will implement docking very soon, and some commercial tools like feature trees from Biosolve IT is going to be one of the first ones. And so you can see you can add as many filters as you want. You can specify the parameters. You can add specific molecules you want to find similar ones to. And yeah, at the end you can click on screen, and here you can see the screen is still running, but you already have found some hits. So as soon as they pass the filters, they will display it on screen. And if the screening is more time-consuming, then you can. So if you go to my collection, you see that it's still running. Already found a couple of compounds, but at this stage you can start doing something else. So for some tools, they used to say that now you can go and grab a coffee or something, but actually at Mcule.com you can start another search. So you don't have to leave the screen as it is. You can just go on, create a new workflow, and do something else. And yeah, so whenever the search is finished, you will get a notification, like this little sign shows you. And you will also get an email notification that your search has been finished. And this is your search result. Yeah, and as I said before, you can export it or you can ask for a quote. And you can export it in different formats. You can generate the file and then you can download it. So this is what it's basically about. And yeah, we will, so the website is just launched. So we will add, so the framework is ready. We are ready to add more compounds and more tools. So if you have specific needs or you want to see a tool implemented in the system, just let us know, and we will try to do the integration. Right. So I will talk about a case study which you can hopefully do in 1 or 2 months on the website yourself if you want. Because all the tools used for this screening will be available. So this work was done actually in a collaboration of Gedeon Richter, pharma company, and our one chemical supplier partner for us, Informatic. And we actually designed a library for GPCRs, a fragment library. So what we have done was we started with the Campbell database, which will be also integrated into the website. So you can grab compounds from Campbell for specific targets. So what we did was we just selected all GPCR, all ligands that have GPCR activity from Campbell. And we did a rule of 3 selection based on the STAX rules, which yielded real GPCR fragments. What we also did was we selected a little bit larger compounds in the drug-like space by rule of 5 filtering. We fragmented all of these and applied the rule of 3 filtering. These yielded virtual GPCR fragments. Some of these are expected to be important for GPCR affinity. Some might not that important, but several interesting fragments are among these. And the last selection was from a commercial fragment library. We did the rule of 3 filtering. Surprisingly, not all the compounds were rule of 3 compliant. But anyway, we did our selection, and what remained from all of these fragment libraries were docked into the recently discovered GPCR X-ray structures. And we applied the consensus scoring, and it turned out that so actually the real GPCR fragments had a significantly higher rank. You can see this in red, while the virtual GPCR fragments had a little bit lower ranks. And this was the commercial fragment library, which wasn't biased against GPCR, so it was just a regular fragment library. So what we did was actually we defined an optimal ranking window, let's say, which is the region where we get a quite nice enrichment for real GPCR fragments compared to random fragments. And we screened the fragment library of our partner, Informatic, and we selected compounds in this region, those compounds that were highest ranked. And so 9 compounds were available from among the virtual hits, and they were tested at our pharma partner on the dopamine T3 receptor. And it turned out that actually they were all active at 100 micromolar, which is a typical screening concentration for fragments. So this gives us a perfect hit rate. And so the best compound, I'm not sure you can see it, but it sits in the binding site quite nicely. It creates a salt bridge with this crucial aspartate amino acid. And it was actually the ligand with the best dopamine D3 affinity. Okay, just to summing up, so what we provide, we try to provide you is a compound database that is up to date and carefully curated. It contains all the purchasable compounds, and you can use this haystack again to do searching and screening on it. You can do simple and complex searches. You can build up workflows from all these tools, and we run the calculations on the cloud, so you can expect the results turning up very quickly. And at the end, if something interesting comes out of this, you can order the compounds from multiple suppliers, and we try to deliver, to aggregate all these and deliver to you as a single package, as an ultimate goal. Right. So yeah, we have just launched and we are ready for beta testers. So we already have quite a few people who enter their email addresses on our website. So I encourage you, if you want to try it out, just go to mcule.com, enter your email address, and in a few days we will send out the first secret links, let's say, for registration, and so that you can try it out. And also we are looking for chemical suppliers for more moleculars, certainly, and software vendors to add your tools, to integrate their tools into mcule.com. Okay, thank you very much for your attention.